Okay, class, this video is part 2 of chapter 3, aerobic metabolism. I'm going to go ahead and share my screen and we'll get started with the lecture. Okay, so our learning objectives or our outline for part 2 is the electron transport chain, which finishes up our aerobic metabolism. Then we'll get into the Cori cycle, lactate shuttle, beta oxidation, and protein oxidation. All right, so first we need to understand exactly where our electron transport chain is taking place. We're still in the mitochondria like we were when we were talking about the Krebs cycle, but we're in a different part of the mitochondria. So if we see here, we are straddling this inner and outer membrane. If you look at our mitochondria, we see the dark orange we know is the outer membrane, and then this orange part here is our inner membrane. So we're straddling that part right there. And if you look at this picture, it will also show or kind of that we're kind of just on or across one of the membranes. So what we're looking at first is a series of protein complexes that are embedded across this inner mitochondrial membrane. I'll explain each of these protein complexes, which you can see kind of as these purple structures here. So I'll explain what those are later. The purpose of the electron transport chain is we know that electrons are captured from NADH and FADH. We have to think back to the NAD and the FAD. Those are carrier molecules, if you remember. Those carrier molecules pick up hydrogens. When they pick up a hydrogen, they're also picking up the electron from that hydrogen. And so a hydrogen is a proton. That's why it has a positive plus, but it has these associated electrons. And so these electrons are also captured as well. So these electrons have to be transferred through these different complexes. And you can see here, some of these complexes are called cytochromes. Give me a second, hold on. Okay, so cytochrome is this hemoprotein or this iron-containing hemoprotein that is mainly responsible for transporting our electrons and help to make us more ATP. Something else you'll see down here is this Q, which is a coenzyme Q. We already know what coenzymes are, but this ubiquinone. This ubiquinone is more so like a mobile transfer molecule of hydrogens and electrons. And so this here is a way for hydrogens, or more specifically our electrons, to get transferred down our electron path. And that's what I want you to look at next. Is in this picture, we have two pathways. We see the blue arrows. These are proton pumps, or they're basically proton pathways. These structures here are pumps that will pump these protons or these hydrogens across from the inner to the outer membrane. And then we see the pink line here is this electron path, and this is where the electrons will travel. All right, so that's kind of the first thing on this slide, and then we'll go to the next slide, and I'll explain a little bit more. All right, so the goal here is to release energy. We want to pump hydrogens from the mitochondrial matrix, which is down here, into our outer membrane, so across this inner membrane space. This is what's going to happen is all of those NADHs and FADHs, and if you count them up, we have 10 NADHs and 2 FADHs. So the NADHs, if you see in this picture right here, kind of in the first pump, NADH is reduced back to NAD and hydrogen. Well, where did that hydrogen go, or that proton? It was pumped through this first pumped down the blue proton pathway. Now we have the hydrogen up here at the top. So as hydrogens move across this membrane, they're actually going to move down a concentration gradient. So the concentration gradient is going to be created when we have, this high concentration, high amount of hydrogens that are across this outer membrane area, this outer membrane space of the or within the electron transport chain. So that creates a higher concentration gradient. So if you continue reading this bullet point, energy will then be transferred to turn a DP into a TP. So we'll talk about that in just a second. So kind of coming back to this picture, all of the NADHs, which we have 10 NADHs from one molecule of glucose, they're dropping their NADH off, or their hydrogen off right here. So they're pumped through the first pump, okay? FADH, for some reason, has to skip the first pump and it is dropped in at the second pump. So over here with this ubiquinone or this coenzyme Q, it's kind of dropped in right here. And so the FADH2 is reduced to just FAD. Well, its hydrogen or its proton is pumped through the second pump, okay? We'll talk about kind of numbers in just a second on how much ATP each of these hydrogens actually make. So we'll get to that in a minute. So purpose of this first part is understanding NADH and FADH, they pump their, or they drop off their hydrogen, they pump their protons up out of this this mitochondrial matrix across the inner membrane into the outer membrane space. What happens to the electrons? Well, it's called the electron transport chain, this little pink pathway. The electrons that are captured when a proton or when a hydrogen is picked up, those electrons will split off right here and they will follow down the electron pathway, okay? So they follow down this electron pathway and are dropped back through the very end. And so this generates energy. So the purpose of this is looking at the energy generated as these electrons are passed down. We also said there's a really high concentration of hydrogens up here at the top. Well, on the next slide, we'll look at kind of a different picture that shows, okay, well, where is all this ATP that we're going to start making? So here we see a similar picture. I like this picture a lot though, because it kind of shows exactly what's happening, exactly what's going on. So if we look at the NADH in this green and the FADH, if we go to this first little thing I have here, complexes of the electron transport chain function to pump their hydrogens from this mitochondrial matrix to this intermembrane space, which is on top here. We see the NADH drop off their hydrogen. It's pumped up here. Here's our proton pump. Same thing, FADH drops their hydrogen off, but in the next pump, pumps up. This creates a high concentration gradient of hydrogens. Our body does not like concentration gradients and likes to even out all of these inequalities. And so what happens is these hydrogens will be traveled and pumped back down through something over here on the far right called an ATP synthase complex. This is where the majority of the ATP is made. So let's kind of look at this one here. This hydrogen gradient is formed, like I just said, with this higher concentration of these hydrogen ions in that intermembrane space, so more so than the matrix. Next, we see the electron pathway. In this picture, it's red. So this red electron pathway is where all these electrons are gathered. If you see this little picture, right, or this little spot right here and these across this pathway, there are electron carriers, and that just helps to carry those electrons kind of all the way across. I'll talk about the purpose of the electrons in just a second. So back to the hydrogens, these hydrogens will then be pumped back down across this intermembrane into something called an ATP synthase complex. And so we know this word ASE means it's an enzyme. So this here, ATP synthase, is an enzyme. We're turning a DP into inorganic phosphate into ATP. We see that right here at the bottom of this ATP synthase complex. As the hydrogens are pumped back through, this generates enough energy to to synthesize ATP. And so the pumping of this hydrogen activates this enzyme, this ATP synthase enzyme, and now we have ATP production. Okay, some other things here is understanding, well, what happens to all these electrons? So these electrons right here occur kind of at this last step. And so if you look down here, kind of at this third pump at the bottom, we see 4 hydrogens plus an oxygen. So this is how we are able to make H2O, water. And so if you remember, I said at the beginning of this that water is the final step in the electron transport chain. It helps to regenerate the cycle and to prevent acidification of the cell, is having these water molecules formed. I also mentioned that oxygen is the final hydrogen electron acceptor. So if you remember, these electrons passing down this electron transport chain are passed back here. Well, these hydrogens that are here will accept those electrons, and we have an oxygen. So technically, if you look at the formula for for water, it's H2O. We know oxygen is O2. So we really just need half an oxygen plus 2 hydrogens for each water molecule. So we have this formula right here. 4 hydrogens plus just 1 oxygen molecule gives us 2 water molecules. And so that's this final step, is we form 2 water molecules and we form ATP here. Again, we want to understand how much ATP are we actually forming. So that gets us to this next slide here. We have to count up everything we've had, and this will help us understand how much ATP we have. Okay, so intermediate step. So glycolysis, 2 NADH, 2 to 3 ATP are produced here. We have 2 NADH from intermediate step. Krebs cycle, 6 NADH, 2 FADH, and 2 ATP. And so after about 3 minutes of exercise, our body has produced this much energy from just 1 molecule of glucose. So that's not enough. We have to make more. And that's what we just learned about the 2 NADH and 2 FADH making more ATP. So what's interesting is 1 NADH is technically worth 3 ATP, but we have to factor in the energy it takes to shuttle the hydrogens across the matrix. That actually spins half an ATP. So we lose half of 1. So 1 NADH, instead of equaling 3 ATP, it only equals 2.5. We had 10, so multiply that. We get 25 ATP from our total, 1 glucose molecule. 1 FADH is technically worth 2 ATP, but again, it costs energy to shuttle these hydrogens across the membrane. And so we lose half an ATP there. So we only had 2 of those, giving us 3 ATP. So we have 28 ATP just from the NADH and the FADH alone, plus the 4 to 5 ATP from all these other steps that we just talked about. So that is a net production of 32 to 33 ATP. If you watch videos, which we will watch videos in here, and if you read textbooks, other textbooks may say we make 36 ATP or 38 ATP. That's the gross production. They're not taking into account the ATP spent shuttling these hydrogens across the membrane. And so that's something to keep in mind, is when you hear other numbers besides 32 to 33, that's this gross production that doesn't take into account the small amount of ATP that we lose with the shuttling of them across the membrane. If we want to see how all of this is made, we can do more calculating here. I like this chart because it shows up here in this first green box on the left corner, ATP, or we see glucose. So within glycolysis, glucose to 2 pyruvates. How much ATP does it actually produce? We have 4 gross production minus 2 that were used, 2 NADHs plus our hydrogens, which are sent over. We then see acetyl-CoA. So pyruvates to acetyl-CoA, what does that make? Makes 2 NADHs. We see the Krebs cycle makes 6 NADHs and 2 FADHs. And then we see our 2 ATP that are produced. So we tally all of that up, and then we do our multiplication. The NADH we know is 2.5 ATP, FADH is 1.5. So we get our numbers, calculate over here. a total yield of 32 ATP if we're using glucose to start with. So this number is something we have to know, we have to memorize. 32 ATP are produced from one molecule of glucose through all the steps, 33 if we start with glycogen. So before we move on, we're going to watch a quick little video that kind of summarizes what we just talked about. So let me share my screen to a different part. We're going to look at a video I have pulled up.
The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
All right, so that summarizes understanding aerobic metabolism with carbohydrate oxidation. So when using carbohydrate as a fuel source, we go through all of those steps, and this is kind of where we end up at the very end. So we do have another topic to go over, which is understanding about lactate. So let me go ahead and share my screen for the rest of this lecture here. Okay, so starting here with lactate or lactic acid, we first want to understand that lactic acid is in a state of constant turnover. So when we think of turnover, we mean changing it into something else or back to lactate. So we can also think of that lactate is lactic acid is highly unstable. I think that I said that before with pyruvic acid, lactic acid, most anything that has an acid associated with it is an unstable molecule, and it quickly disassociates from the acid form and turns back into lactate with a hydrogen. And so when we refer to the term lactate, lactate is mainly produced in type 2 muscle fibers, which are more of our fast twitch muscle fibers, but they're transported more to our aerobic muscle fibers, which are type 1 fibers. Lactate will then be taken up within the mitochondria of our type 1 fibers, which is where they're shuttled to from the type 2 fibers, and they're oxidized. So we want to understand this term oxidized if we don't know what it means already. So it's undergoing a reaction where electrons are lost to form a specific product. So we've talked already in this class about a lot of reactions that occur. We've talked about oxidation. When something's oxidized, we're basically going through a reaction, we're losing electrons. All right, so we want to understand 2 different ways in which lactate is transported away from the site of production. So one thing I want to make clear is the most common, I guess, way that lactate is used is taken up by the mitochondria, which is this first or the second statement right here. So this second statement about lactate taken up by the mitochondria, this occurs most often. This is how lactate is cleared, is it's just taken up by the cell that produced it. Two other ways of transport are through something called the Cori cycle or the lactate shuttle. So first, let's look at the Cori cycle. Cori cycle is also called the lactic acid cycle. Just for reference, I call it the Cori cycle. So Cori cycle takes place in the liver. And purpose of this is when lactate's produced via glycolysis, when we take pyruvate, we turn it into lactate, we move it to the liver and we're basically turning it back into glucose. So what I want you to do is start here at the bottom for this little flow chart with the blue arrows. From high intensity exercise, we produce lactate. Lactate can be turned back into pyruvate and back into glucose. How though? It's done in the liver here. And so it can be stored within the liver as glycogen, or once it's turned back into, so lactate, pyruvate, glucose within the liver, it's then transported as glucose in the blood, and it can go back to working muscle. So it can go back to working muscle and it can be stored as glycogen within the working muscle, or it can be used for glycolysis through the, to make more ATP, which is just kind of what we learned. If we see down here, Cori cycle in this picture, in the left corner, we see fast glycolysis. At the bottom, look at our skeletal muscle, glycogen to glucose, glucose, fast glycolysis, we make pyruvate, makes lactate. Well, lactate travels in our bloodstream, and it travels to the liver where lactate is turned back into pyruvate, turned into glucose, where it can be stored as glycogen within the liver as a fuel source for later, or that glucose is transported back in the bloodstream to the working muscle so we can use it for fuel. So what's the purpose of this process? To prevent something called metabolic acidosis and to produce more ATP. Metabolic acidosis is sometimes the term is used interchangeably with another term called lactic acidosis, but this is more of the correct term. With metabolic acidosis, it is referring to a buildup of lactate within the body. So when we start exercising, lactate levels immediately start to rise. So lactate levels rise, and we'll look at in part 3 of this lecture how we hit a certain point and our blood lactate levels start to accumulate in really high amounts, which can cause fatigue. So we want to prevent this metabolic acidosis or this buildup of excess lactate within the body, and turn lactate into a fuel source. And so that's what the purpose of this Cori cycle is. It takes the lactate that was not metabolized by the cell that it was produced in. So whatever lactate that wasn't used or metabolized or oxidized is taken to the Cori cycle, or it's taken through something called the lactate shuttle. This is a hypothesis, which means it is like not completely proven yet. We now just have a hypothesis of what else happens to lactate. So it describes movement of lactate within cells and between cells. And so we'll explain what that means. And so we already went through the blue arrows, right? So I want you to look at the red arrows. So skeletal muscle, high intensity exercise, we're exercising really hard, doing some intense work in our short time frame during glycolysis. Glucose turns into pyruvate, turns into lactate. Lactate gets into the bloodstream. And then lactate has two options. We can go through the Cori cycle, which is our blue arrows we just went over, or we follow the black arrow. So this is this first bullet point, lactate moves within cells and between cells. Well, it can be transported to inactive muscle. It can be transported to heart, to the kidneys, where it's turned in from pyruvate into glucose, and it can be used as a fuel source. So basically, purpose of the lactate shuttle is it is another way to fuel our organs. And so the brain, the heart, the kidneys, inactive muscle can take lactate and use that as a fuel source. So like I said, these are our two other pathways in which lactate can be used by the body. So lactate is produced in the cells, and if it's not taken up by the mitochondria and oxidized, then it goes through these two pathways. All right, our next topic is understanding beta oxidation and protein oxidation. If you remember, we looked at the Krebs cycle and I told you to ignore the amino acid arrows for protein oxidation and the beta oxidation fatty acid, like ignore that. Well, here we are now to talk about it. Okay, first being beta oxidation. So beta oxidation, we'll talk about the process of taking fatty acids that are stored like as triglycerides, we break them down and turn them into the 3 fatty acids. They're transformed into acetyl-CoA directly. So we'll discuss this process first and talk about how the fatty acids go through the process of being hydrolyzed and broken down into acetyl-CoA, so then they can enter into the Krebs cycle to make ATP and go through the same process we just learned. Next is protein oxidation. Protein oxidation, we have amino acids that are turned into pyruvate, where they can go through or turn into acetyl-CoA, enter into the Krebs cycle directly. And then, so go into pyruvate or enter into the Krebs cycle directly. Okay, so here we go, starting with kind of reviewing the breakdown process of triglycerides. So we know the enzyme hormone-sensitive lipase breaks apart the triglyceride that's stored in our fat cells or adipose tissues or adipose sites. So we are breaking them down into glycerol and a free fatty acid. So this process we know as lipolysis. Within the adipose site, so here's our triglyceride, we see hormone-sensitive lipase. The reason it's called hormone sensitive is because it is impacted by hormones. We see down here epinephrine. We go through this process with cyclic AMP and something called a protein kinase to activate hormone sensitive lipase. I'll discuss this process down here a little bit more in chapter 8, which is our hormone lecture. But all you need to know about our lipase is our enzyme. It splits the triglyceride. So now we have free fatty acids in the glycerol molecule that have been transported into the blood. Our cells are going to take up the lipids. So we break it down from the adipocyte and it's transported into like skeletal muscle cells. And it is used in aerobic metabolism. We look down here and see that free fatty acids will enter into those muscle fibers by diffusion. So if anyone knows what diffusion means, so diffusion is moving down a concentration gradient where we can easily cross a permeable or a semi-permeable membrane. And so that's how the free fatty acids enter into the muscle. The rate of entry depends on the concentration gradient. So it depends on how strong the gradient is. If we have high amounts of fatty acids already within the muscle cell, then our concentration gradient is relatively stable and we will see a decreased rate of entry or rate of diffusion. But if there's higher amounts of fatty acids outside of the muscle cells, so that we have like lipolysis has been occurring, and we have low levels of fatty acids inside the muscle cells, that creates a greater concentration gradient to where we see increased diffusion or rate of entry of the fatty acids into the muscle fibers so we can use them for aerobic metabolism. Okay, so here we'll talk about beta oxidation. And so there are several steps that take place in order for a fatty acid to be broken down. First, it has to be activated. So we have to have, and you see over here on the right side, the free fatty acid is within the cell, but it's outside the mitochondria. We have this activated fatty acid part. So the purpose is to turn fatty acids into acetyl-CoA. And we have several steps to do that. I'm not going to spend time like really going into this activated part, but we know usually when something becomes activated, it's activated via an enzyme. And so we have this enzyme activation of this fatty acid, turning it into fatty acetyl-CoA, and then fatty acetyl-CoA turns into acetyl-CoA. This process, though, requires 2 ATP per fatty acid to start, okay? So 2 ATP per fatty acid to actually start the process, which is very similar. This is not unique. This is what happens when we start with glucose or glycogen. We have to invest ATP into glycolysis in order to make ATP. Same process here is we are starting the system with our 2 ATP. So we've lost 2, we've invested 2. But the process, this short or small process here, will generate 1 NADH and 1 FADH. We can take those hydrogen electron carriers and we can use those in the electron transport chain to make more ATP. What we want to understand is the amount of ATP produced depends on the specific fat that's oxidized, but always fat will generate more ATP than glucose. Let me give you an example. So the amount of ATP produced from a fatty acid depends on the amount of carbons that are contained within the fatty acids. Glucose molecules contain 6 carbons. So glucose is a 6-carbon molecule. Fatty acids contain anywhere from like 14 to 24 fatty carbons. So 14 to 24 carbon molecules compared to the 6 carbon molecules from a glucose. So that is why fat produces more ATP. This process is a lot slower, though, just like we've discussed. So fat breakdown or fat oxidation goes at a slower process. So it takes longer to start up this beta oxidation. Even though we do get more ATP in the end, the rate at which the ATP is delivered into the cells is a slower process. All right, so kind of next topic here, we're going to talk in a second and get into the protein oxidation, but we want to answer this question first. All right, so we're looking at the production of lipids from glucose and amino acids. So we just talked about, okay, we're using we're using fatty acids for fuel, but what about turning a fatty acid or making a fatty acid from other sources like glucose and amino acids? So question, can we gain fat mass, like stored fat on our body, if our diet only consists of protein and carbohydrates? We look at this picture, which is not the best picture. There's one on the next slide that I like better, but it basically shows how amino acids can be turned into pyruvate or acetyl-CoA. Acetyl-CoA, we already know, can directly turn into fatty acids. We just learned in beta oxidation that fatty acids become activated and turned into acetyl-CoA. Well, that process can also happen in reverse. So we can have acetyl-CoA turn into a fatty acid. We know fatty acids are stored as triglycerides. So same thing here, pyruvate can turn into glucose, which can turn into glycerol, which makes up our fatty acids. So basically, whatever we consume, protein, carbs, can always be stored as fat mass. And like I said, this picture I like a lot. So if we look up here, we have our food intake. We have fats, carbohydrates, and proteins. So let's look over here and start with protein. We know protein is broken down into amino acids. Amino acids are technically not stored on the body. It contributes to our body's protein, which is our muscle tissue found in bones, skin, hair, all of that. So it goes to our body's protein, and we have protein breakdown, which contributes to our amino acid pool, which is in our bloodstream. If you follow the dotted line, it's a minimal contribution to metabolism. We don't actually want to use them for energy. So the amino acid pool can be turned or used back for protein synthesis, goes back to the body's protein, or it goes through a process called gluconeogenesis, which we did learn that term a while ago, and we'll bring it up one more time. Gluconeogenesis is taking the amino acids and turning them into glucose, which goes to our glucose pool, okay? So now let's start up here at carbohydrates. Carbohydrates we know turn into glucose. Follow the first arrow. Excess glucose goes through lipogenesis, formation of fat, which fat is stored on our body. We know also glucose goes to our glycogen stores, so we can store it as glycogen. We can go through glycogenolysis to make our glucose pool, which is our blood glucose or blood sugar. It can go back to glycogenesis, which is the formation of those glycogen stores. But we know that glucose pool, our blood sugar, contributes significantly to our metabolism. And then let's look over here at fats. Fats broken into triglycerides. So free fatty acids and our glycerol. We already know what happens to the glycerol, but our free fatty acids can go to our fat stores directly, or they can go through lipolysis to our free fatty acid pool, which is in our bloodstream, which was the process we just learned about that goes to metabolism, or lipogenesis back to our fat stores. We should know every single term on this slide. So every single term on this slide, you should be familiar with. And you should understand this entire process right here of how carbohydrates and proteins can be stored as fat when we consume them in excess. All right, next is continuing with that discussion about gluconeogenesis. Anything that's gluconeogenic is making glucose from a non-carbohydrate source. And so we do that with lactate. We do that with glycerol. And here we're going to show two ways we do it with amino acids. Okay, we have two terms. Basically, this first term, transamination, is taking apart the amino group and the acid group. And so what we're doing with the amino acid is our amino group is transferred to something called a keto acid. A keto acid is an organic compound that contains a carboxylic acid group and a ketone group. This keto acid here is used to form new amino acids within the Krebs cycle. And this takes place within the liver first, and then transported back to skeletal muscle where we can use it. The next type is deamination. Whoops, let me go back. Deamination is when our amino group is removed and recycled. So when we say recycled, it just means the components of that amino group were either turned into glucose or pyruvate or acetyl-CoA. So basically taken apart and recycled by the body so we can use it to make something else. The acid group is converted to ammonia. And so that's what this picture on the right explains or shows. We have our acid or our amino acid, the amino group's removed, and the acid group is turned into something called an ammonia. So ammonia is a base that can upset the acid-base balance in the body. When we say upset the acid-base balance, it means cause more acidity, cause the pH level of our bloodstream to drop. To prevent this, what's going to happen is ammonia and carbon dioxide combine together, and that forms urea. So urea is how a lot of the nitrogen is excreted by the kidney. So this is just a compound found in urine. And so urine is really the only way to measure the amount of protein that has been metabolized or catabolized by the body. Which again is not something we actually want to happen. We don't want to use protein or amino acids as a fuel source, but we can measure this by measuring urine samples and seeing how much nitrogen has shown up within the urea within our urine. Because we know nitrogen is not found in fat and it's not found in carbohydrates. It's only found in protein. And so nitrogen in the urine can determine how much protein the body is using. But a healthy body typically does not use amino acids. If they do, it's less than 10% of your ATP comes from amino acids. And so the transamination, deamination process, they occur within the liver, and then the deamination occurs also in the kidney mitochondria. But this is rare, I'll say, and it only occurs in a few instances like these right here. So we know amino acid metabolism, very small, 10% or less, except when we are extreme dieting. So when we go through an extreme diet, and when I mean that, I mean cutting your caloric intake to less than 1,000 for someone who needs at least 2,000 calories a day. So cutting your calories it forces your body to find other fuel sources. If you're not consuming enough carbs or fat to fuel your energy systems to give you energy, then your body has to turn to muscle and starts breaking down your muscle tissue. And that's where the amino acid metabolism comes in. Another is kind of the opposite. It's like a high protein diet. And so let's say that most of your diet consists of protein. Well, our body's really good at finding what it needs, and it's going to do this the easiest way. And if we have this high availability of amino acids in our bloodstream, in our amino acid pool, it's going to take that. And so it's going to use what's most available. And so if amino acids are most available, it's going to take them and metabolize them for fuel. And so high protein diets. And then long-term endurance activity will activate an enzyme, proteases, protease is a enzyme that breaks down protein within the muscle tissue. So think of exercise that lasts for 2+ hours, 3 goes all the way 5 hours. Long-term endurance activity could be anything from jogging to hiking, cycling for long distances. Our body would start to turn to other fuel sources to give us energy, so breaking down muscle. Okay, last part of this lecture is looking at a research study. So in this study, 12 weeks, the men were average age 20, and they lost on average 20 pounds. But we have 3 different groups. We have a dieting group and a diet aerobic exercise group, and then a diet aerobic and resistance training group. They all lost weight. They all lost about 20 pounds. But those percentages that you see for each group determines exactly how much weight came from fat tissue. So first group, the diet group, 69% of the weight they lost was from fat tissue. Okay, so where did the other 71% of the weight they lost come from? It came from muscle tissue, okay? Think of 100%. Well, only 69% of the weight they lost was from fat. Well, where did this other small percentage of weight they lost come from? Muscle. We had aerobic exercise, which we know aerobic can be weight-bearing or somewhat weight-bearing. And we're using our muscles during aerobic exercise. Well, now only 78% of the weight they lost was from fat. So we increased that bar, that percentage. So of the 100% of weight they lost, 78% was from fat. Okay, well, where is the other 22% of the weight they lost come from? Still, it came from muscle. We preserved a little bit. But now we do the same thing, but we add resistance training. So our last group, 97% weight lost. So now almost the entire 100% of my 20 pounds came from fat. Only 3% of the weight I lost came from somewhere else. It came from muscle. So what conclusions can be drawn from these findings? Hopefully you get it. If we add resistance exercise, resistance training, weightlifting, that helps to preserve muscle when trying to lose fat mass. So when trying to lose weight, dieting alone also loses muscle tissue. Diet aerobic exercise helps to preserve muscle a little bit, but adding that resistance training helps to preserve your muscle because your muscles are going through anabolism. They're building up or they're being used constantly, so they're staying the same. We're not seeing protein taken from the muscle to be used in metabolism. So that should be your conclusion there. But that is it for part 2. So we have completed part 2 of aerobic metabolism. If you have any questions, please let me know, and I will send an email with updates on kind of what to do next or what to expect.